you can take the time to chat about
to have an opportunity to present this work. So this work is something that I did now about 2 years ago with Professor Mukul Bansal at UConn while I was a master's and undergrad student there. And I'm happy to now get a chance to present this work to this audience. And happy to talk about the implications for applying this to problems in the field. So as we jump in, this is a brief overview of the topics that or the general layout for the talk today. So we'll start by covering some background about what the problems that are relevant, that is relevant to this simulation framework in the field of phylogenetics, and then moving into the methods itself about what we do to kind of implement this simulation framework, and finally about applications of problems that this simulation framework can help us solve, and about benchmarking different methods on phylogenetic problems using this simulation framework. So to start things off, in this line of work, we generally are interested in looking at evolutionary trees and how gene families specifically evolve inside species trees, and later on, how other gene domains evolve within gene and species trees. So what we mean by that is, if we look at a species tree, such as the one on the left, we can see that it shows how these different species share this evolutionary history, where species A and B are most closely related, and the other species are more divergent as they share a recent common ancestor that's much higher up in the species tree, indicating they're more distant in terms of their lineages. And within this group of species, we have examples of different genes which might be evolving through different events, such as gene duplication, horizontal gene transfer, especially in microbes, and gene loss, which results in a gene tree for any given gene to look quite different, such as the one that we have on the right here from the species tree, due to all these different events and rearrangements that give us a different gene tree topology compared to the one that we have for a species tree. So when we reconstruct, let's say, a species tree and a gene tree given the biological data that we have, the information that's often missing, and that is missing in these cases, is that we don't know exactly which evolutionary event happened at these different nodes of the gene tree that resulted in this particular gene tree to evolve down this given species tree. So given these inputs, one of the problems that we're interested, especially in phylogenetic reconciliation, is to come up with an example of this gene tree which fits inside the species tree, which can essentially explain what different evolutionary events happened along the course of evolution for the specific gene, such that it has the current, it yields the current tree that we observe with the given data. And this is, I guess, an overview of phylogenetic reconciliation with gene trees and species trees. Now, this can be further extended to gene domains, which now evolve inside multiple gene trees, which then would evolve in turn inside the species trees, which is kind of looking one step deeper into this problem of different trees evolving inside each other. And this is also a problem that we have worked on in Professor Bansal's lab, and there's some interesting work in that area. So the need for simulation really comes from the fact that there is an absence of biological data regarding the specific events that have happened back in time. So when we have, for example, a phylogenetic reconciliation method or some method that tries to infer these evolutionary events in the history of this specific gene family or this gene domain, then it's difficult to actually evaluate whether or not that method is actually giving us the accurate results, because the ground truth, so as to speak, is kind of missing in this sense. So simulation in that context provides us with a powerful technique to actually give us some semblance of what could possibly be a set of evolutionary events that could have happened that leads to a specific set of species, gene, and domain trees that then, if you now test, if you run the algorithm on those trees, you can then check whether or not that matches the actual events that that were created by the simulation framework to produce those trees. And if the simulation framework can accurately model how these species, gene, and domain families are evolving, then we could assume that an algorithm which picks up these events accurately reasonably can model the evolutionary history of these genes and species and domains accurately as well. And there has been work that has been done in this area already. So SaGePhy is work that does something similar, which is it simulates the evolution of species trees and locus trees, and then within loci, it evolves gene trees. And this supports multiple different evolutionary events, including duplication, loss transfer, conversion, and incomplete lineage sorting. So this supports a host of different other methods or other evolutionary events. And it implements replacing transfers, but not additive transfers. So whenever there is an instance of a gene transferring from one species to another, it replaces the existing lineage at the recipient instead of creating a second copy of that specific gene in the recipient species. And it allows for distance bias in choosing transfer recipients, so such that recipients for specific gene families that are further away on the tree would be biased against receiving transfers from the donor, as that would accurately model our understanding of gene transfer events. And this is generally, one of the more advanced simulation frameworks that's available, and we definitely want to take some of the ideas here and add the ideas regarding domain transfers and a few other things to what is already kind of existing in this framework. And there's also another framework which is called Prime Gen Phylodata. This supports a two-tree construct where they evolve a guest tree or a host tree, which kind of relates to is analogous to our species tree, and so a host tree and a guest tree, which is analogous to the gene trees that in our case. And this simulation framework supports gene duplication, loss, and transfer, not the other events that SaGePhy also supports. But essentially, these are the set of events that relate very closely to some of the phylogenetic reconciliation work and some of the other evolutionary inference work that that are methods that we want to test align closely with. So in that sense, this is good to kind of get a basic set of features that we can build up from. And this implements additive transfers, but unlike SaGePhy, does not simulate replacing transfers. So that is kind of one of the limitations here. And in addition, it also does not allow for distance bias in choosing transfer recipients. So, but neither of these simulation frameworks that were available when we constructed this implement both replacing and additive transfers, which are both events that are important in the evolutionary history of gene families, especially in microbial species. And neither allow for starting the evolution of a gene tree from somewhere other than the root of the species tree, which is something that can definitely happen in a lot of cases, as not all gene families might be present at the very common ancestor of a certain group of species. And neither also support subgene level events, such as partial gene transfers and in addition to that, neither of them support protein domain evolution, so which is essentially evolving a domain tree within multiple gene trees, which itself evolves down the species tree, which is one of the main additions that we have in this simulation framework. So our contributions in that regard is that we build up from Gen Phylodata, which is essentially a more basic, simple model that we can implement additional features on top of. So we implement both replacing and additive transfers, and also with distance bias when selecting transfer recipients. We allow for starting the evolution of the gene tree from somewhere other than the root of the species tree. And also we support subgene level events. And in addition to that, we also support protein domain evolution. So allowing for the evolution of domain trees within multiple gene trees. And with that, let's kind of jump into the methods. So, what is kind of like the basic ideas behind these simulation frameworks? So GenPhilo data itself consists of these two independent tools, as I alluded to before, which concern the simulation of phylogenies. So there's essentially generating trees. So with HostTreeGen, this tool generates a species tree, and GuessTreeGen, which generates what we can find an analogy to would be the gene tree, which is it generates another tree which is guided by the framework already created by the first species tree that we have generated. And both tools use something called the birth-death process to generate trees, which kind of models how lineages are created and destroyed as a function of certain rates of deletion and speciation or duplication events. So the basic idea is that, the different states or the numbered circles here correspond to different lineages in a specific tree. And with a certain rate lambda, you would have additional lineages being created. So as a tree gets evolved from the root all the way to the leaves, you would have, at a rate of lambda, you would sample from a distribution which samples either duplication or speciation events, which create additional lineages, and at the same time, you would also sample deletion events, or essentially events that would reduce the number of lineages, which would go back, which would in this case be mu. And the idea here is that with, for example, HostTreeGen, which is the species tree building method, you would start off with a stem at the beginning, and you would, given these different rates, you would evolve, you would sample different evolutionary events along the course of this tree from the top to the bottom. And at each of these events, you would either have a speciation or deletion event, which would either add more lineages or prune away, or essentially stop the evolution of lineages which correspond to deletion events. So if you were to build a tree using this, you would provide it with a speciation rate and a loss rate, and essentially a time interval which would correspond to how deep you want to generate a tree for. And using these, the framework would essentially sample from an exponential distribution using a sum of these different rates, which would roughly correspond to what is the how many, the higher this rate would be, that would correspond to sampling more events across this time interval. And for every sampled event, it could either be a speciation or a loss, which would depend essentially on the ratio between the speciation and loss parameters that you give it. And you can also specify other things like the minimum or maximum number of leaves that you want to generate, and the tree would and the software would essentially generate different trees until you fit the constraints, essentially. And with GuessTreeGen, which is the gene tree generating method, what the current framework supports is essentially evolving another tree inside this already species tree that we've created from before, which evolves in a similar manner, except the fact that whenever you hit a speciation event, which is kind of the dark gray ovals here that you can see, then that automatically results in this the speciation event in the or this duplication event in the or not duplication event. You have this second lineage that gets created in the gene tree, which corresponds to having a lineage evolve down either of the two children in the species tree. And in addition to that, you also sample events just like you did in the species tree, for which you would either which would either correspond to duplication or loss or a horizontal gene transfer depending on the ratios of the different rates for each of those events that you provide the framework with. So this would correspond to essentially giving it a duplication, loss, and transfer rate, and also a species tree down which you want to evolve this tree. And again, the sum of these rates would correspond to how often you would sample, or how frequently you would sample these different evolutionary events. And for each sampled event, similarly to before, you would have a probability of that event being a duplication or a loss or a gene transfer, depending on the ratio of that specific parameter compared to the sum of the combined set of the different parameters that you give it. And for using these, you can control, for example, how frequently do you think in a particular scenario that horizontal gene transfer is prevalent. Maybe you want to, if you set that to 0, you would only have duplications and losses, and so on and so forth. So if you can match the biological scenario with the parameters that you think would be appropriate, then you would essentially, in theory, be able to generate a tree which accurately reflects the evolutionary scenario in a particular biological data set. And in terms of the additions that we make to GenPhilo data to further improve the distance, the transfers, we implement distance bias transfers, which essentially reflect the fact that we observe in our data that, and especially data from our collaborators, which shows that the frequency of horizontal gene transfers decays exponentially with the evolutionary distance between lineages. So that is what we want to reflect in our software. So whenever our improved version of GenPhilo data samples species candidates for transfer, instead of doing it uniformly at random among contemporary species, we would do this in such a way that it reflects a reality, which would be that we would assign weighted probabilities to the potential candidate recipients as a function of their phylogenetic distance from the donor. And users can, when using this, choose multiple different options, including options such as having no bias, which would be essentially what was covered before in GenPhilo data, having a bias that essentially increases inversely proportionally to phylogenetic distance, and one that exponentially decreases with phylogenetic distance, which would reflect, and which is the default and reflects the patterns that we observe in the data. And for the exponential option, users can define a rate parameter, which essentially would correspond to how sharply you want this exponential decay to be as the phylogenetic distance increases between different transfer candidates. And of course, yeah, and with this rate, you would sample this probability from an exponential distribution of any given number of any specific recipient receiving the transfer from a given number of potential candidates. And we also, what we improved from both GenPhilo data and SaGePhy includes this location of gene birth addition, where we instead of evolving the gene tree directly from the root node of the species tree, we essentially assign weighted probabilities to all the different nodes in the species tree that exponentially decrease as you go down in the height of the tree, which can be used to sample different starts of specific gene family evolution within the species tree, which would more accurately reflect what would happen in evolutionary history for a given species, set of species and gene family in reality. And these weights are, of course, biased towards the root of the tree, as otherwise you would end up with really small trees that mostly are near the leaves of the species tree. And one of the biggest changes here, or the additions that here to both of the simulation compared to both of the existing simulation frameworks is the domain tree gen, which we add on top of the existing framework, which allows for evolving a domain tree on top of or within multiple gene trees. So this would support events exactly like we have for the gene tree, such as domain duplication, loss, and even transfer. And this would take in a species tree and a set of gene trees, all which have evolved down the species tree. And it would allow for transfer events across both gene trees and within different species within the species tree. And you can also enforce the evolution of this specific domain across all given trees, across all given gene trees within the set of gene trees that you provided with. And this essentially picks a random gene tree to start the domain tree evolution, and using the parameters that you give it, just like before, it evolves a tree that covers or essentially evolves down all these different gene trees, which in turn evolve down species trees, which would give you now a way to now further evaluate methods which look at domain tree evolution and reconciliation with gene trees and species trees. And using kind of leveraging this this ability to generate domain trees, we also are able to implement partial gene transfers. So essentially, in a partial gene transfer, parts of genes are transferred between different species instead of the entire gene. And we can use a tool like SeqGen, in this case, to evolve nucleotide sequences down this simulated gene and domain trees. And this would allow us to essentially take advantage of the gene to leaf mappings, the gene leaf mappings between the domain trees and the gene trees to essentially either randomly insert or append these shorter domain sequences that we evolved down the domain tree into the longer gene sequences, which would then reflect partial gene transfers, essentially because the domains are subgene level units, which also undergo transfer events as part of the framework, which would allow us to simulate this effect of transferring parts of different genes down this phylogeny. And with the partial gene transfer framework, you can provide it with a set of domain trees that you generate and a set of gene trees that you generate. And in addition, the domain to gene leaf mapping, which is generated by the simulation framework. And you can specify the length of the domain and gene sequences and any substitution models that you want to use. And this results in a seamless transition from the simulation framework itself in terms of the outputs that it generates to then easily use those to come up with these partial gene transfer scenarios where you can get these sequences of genes with embedded subgene transfer level events. And this would produce sequence files for gene and domain trees in faster format, which you can then run your appropriate methods on, and then use the known somewhat pseudo ground truth generated from the trees to test whether or not you can recapture these subgene level events using the methods that you apply to it. And that kind of brings us to the application of what of potentially what are some of the different things that we can apply such a simulation framework to, and look at some of the applications that we've already done to test out some of the methods that we use in this space. So one of the methods that that we use in terms of studying the evolutionary history is detail reconciliation, so duplication transfer loss reconciliation, which is generally mainly used to study microbial evolution. And it's, though it can also, of course, be used to study any kind of scenario where transfer events are play a role in the evolutionary history. And generally, a detailed reconciliation is formulated using a parsimony framework. So you assign costs to these different evolutionary events. And since there are multiple different assignments of events to a gene tree topology, which would reflect an appropriate correspondence to a given species tree topology, we would try to find a set of different evolutionary events within this gene tree that correspond to the lowest cost when fitting in this gene tree within the species tree. So currently, the DTL reconciliation framework itself assumes that all the transfers are additive transfers. And the problem actually has been shown to be NP hard if both additive and replacing transfers are considered simultaneously. So what we want to actually see if when we find this reconciliation of a gene tree which evolves with both additive and replacing transfers, which our simulation framework supports, then can the reconciliation also work equally well? So that's essentially the objective here. So we will evaluate whether or not the DTL reconciliation framework, in the presence of both additive and replacing transfers, as simulated by our framework, can capture these evolutionary events, which is, can it capture the duplication, transfer, and loss instances without classifying between replacing and additive transfers? Can it still capture the positions of those different events within the tree, and whether or not those evolutionary events are labeled as appropriately, like just transfers or duplications or losses? So what we would do in this experimental setup is we would simulate different species trees and gene trees with both additive and replacing transfers. And again, the DTL reconciliation framework cannot tell the difference between the two, but we want to see if being agnostic of the two types of transfers will affect its performance or not. And we want to then use this DTL reconciliation framework as implemented in Ranger DTL, which is a software package for doing so that is developed by Professor Bansal in our lab. And we want to see if this framework, which assumes that all transfers are additive, how well this performs in inferring the different event types and their mapping on the species tree without being aware of replacing versus additive transfers. So we simulate 100 species trees, each with 100 leaves, and we generate 3 sets of 100 gene trees for each of these different settings, where we have trees which are evolved with low DTRL rates. So this would stand for duplication, transfer, and replacing and additive loss. And also with medium and high rates of these different events, to essentially test whether or not the method does poorly or better in a certain setting where there's a high frequency of different evolutionary events happening. So with the simulation, we generate the set of trees where the average number of leaves for the low, medium, and high set of DTRL rates, of course, correspond roughly to, well, for the high DTRL rate, we have a large number of leaves, though between the medium and low, there's the trend is not exactly linear. But for the number of events in general, we see that as the DTRL rate increases, we essentially sample more events of not only duplications, but also additive and replacing transfers as the rate of these different events that we provide the framework with goes up, which would essentially reflect our expectations with this DTRL rate. And then we look at the accuracy of this Ranger DTL algorithm in first picking up the different events. So essentially this what this plot shows us is what proportion of the different events which are actually speciation, duplication, or transfer within the simulated tree are captured by this Ranger DTL framework. And we see that the vast, in fact, most and nearly all of the different speciation, duplication, and transfer events are, as shown by the left-hand side chart, picked up accurately, essentially labeled correctly in this tree at different rates of evolution by this DTRL framework, which again is not aware of these two different types of transfers going on. But regardless, we see that it nevertheless, captures these different transfer events, and it also maps these different transfer events happening in the gene tree accurately to the positions on the species tree, which would reflect at which point in the evolutionary history of a given set of species these different events have been occurring. So this shows that the DTL reconciliation framework is fairly robust, not only to the different rates of these different events, even though we see a slightly lower mapping accuracy with the higher rates, we generally see that it's fairly robust to these different rates and also to the presence of the two types of transfers. And looking more closely at the rates, at the accuracy with regards to picking up both replacing and additive transfers. Now, again, as a reminder, range of DTL will not say whether or not the event is a replacing or additive transfer, as the method is agnostic to that. But even without essentially, having that knowledge, range of DTL can pick up or essentially accurately label transfer events of either type as transfer events, regardless of the DTRL rate. And it also does a fairly good job at mapping those transfer events accurately onto the species tree. So this is, I guess, in one way, a nice validation of the fact that we can pick up these different evolutionary events, even in the presence of these different transfer events, without specifically modeling these differences in replacing and additive transfers, which this simulation framework kind of allows us to do by providing, in a way, this ground truth in this simulated species and gene trees. And going further, this SaGePhy method has also been used in published research from our lab to essentially generate simulated gene trees with both additive and replacing transfers, along with the other evolutionary events, and with different branch scaling schemes to then assess whether or not to essentially assess the accuracy of different gene tree rooting methods, which is also one of the problems in this field of essentially, given a gene tree, which is how do you root that specific gene tree, or essentially, where in that unrooted tree that you construct from a given set of sequences is the common ancestor, essentially, of all these different leaves of the tree, and in which direction would essentially time be oriented in the evolutionary history of this of an unrooted gene tree. So this is work that again applies to the simulation framework, and also recently work that was published at ACM-BCB 2020 is work that uses SaGePhy to generate simulated gene trees with again different rates of in this case different rates of additive and replacing transfers along with the other events. And this tests different reconciliation-based heuristics for now classifying the inferred transfers, which we know range of DTL, which doesn't differentiate between the two types of transfers, does well at essentially labeling them as transfers. But now going further, this paper now essentially tests these different reconciliation-based heuristics for classifying these inferred transfers as being either replacing or additive, which now our simulation framework can allow to test, since we simulate both types of events. And in this work from our lab, Abhijit and the co-authors, they train a supervised machine learning framework to essentially classify these inferred transfer events as additive and replacing, and they impressively show that the ML model actually outperforms other heuristics that exist for classifying transfers as replacing or additive. So this kind of just shows that this simulation framework can be applied to lots of different problems in this field of inferring evolutionary histories of genes and species, and of course now domains. So in conclusion, we developed the first simulation framework with support for both replacing and additive transfers, which allows us to essentially combine some of the useful features that exist independently in other methods, and also on top of that add other features which are useful for testing different hypotheses, which include simulating subgene level events such as domain evolution and partial gene transfers. And using this, we can now show that DTL reconciliation performs well at recovering both types of transfers, even without knowing how to tell the difference between the two. And we also show examples where we can now use the generated phylogenies using this simulation framework to test methods in the field on inferring evolutionary histories of a given set of genes and species. And of course, this makes possible for work to be also done on domain level events. And with that, I would like to feature some acknowledgements. So of course, big shout out to the Comp Biolab at UConn, led by Professor Bansal, for tremendous support and all the guidance that has made for a great time when I was there for my undergrad and my master's, and to some of my other advisors as part of the University Scholar Committee at UConn, which include Professor Mondoyan, Professor Gogarten, especially for his expertise with horizontal gene transfers, and of course to these National Science Foundation awards, which support this research. And that reaches the end of the presentation. So I'll take any questions and we can have a discussion. And thank you.
Thank you very much, Shomo. That was a very interesting presentation and very clear. Also, you went deep in the methods and also in the description of the application. So are there questions from the attendees? You can raise your hand or ask your question in the chat. I will also open the chat if you have questions. Otherwise, I can start with my own questions. So, yep. So I understand that you made a distinction between partial gene transfer and domain transfers.
Yes. Mm-hmm.
Okay. Mm-hmm. So yep. It's not the same. So is it that for partial gene transfer, you also have the replacing and the additive distinction between the kind of transfers? Do you also have that?
So the partial gene transfers essentially is modeled as using the domain level transfer events. So you can essentially, using this framework, you can have a separate tree which models specific domains. And with the partial gene transfer, essentially is essentially a extended feature of the domain transfer itself, where using the domain tree, you then evolve sequences down it, right? Which can then be embedded inside the gene tree. So with the partial gene transfers, the different features such as transfers and duplication and all these events are modeled inherently by the evolution of the domain tree, which goes through these different events. And what that gives you essentially is a set of sequences within the gene tree, which are augmented with the domain sequences, which essentially give you information about subgene level events, which kind of essentially just, I guess, builds on top of the domain tree features.
Okay, thank you for the answer. So I have a question in the list of questions. So there is a question from Cristal. Cristal from the Federal University of Rio Grande do Sul, and she's asking if she can analyze repetitive elements and gene evolution using this tool.
Can you analyze repetitive elements using this framework? Well, certainly. I mean, I think this framework is agnostic to the sequences that are concerned here. So essentially, this framework only generates trees. So if you want to now have a substitution model which favors potentially repetitive elements, And again, I guess I'm certainly not an expert there, but yeah, like this framework is completely agnostic to sequences. Sequences are only evolved after the trees are constructed. So if you have a specific model for coming up with substitution models for sequences, or if you start off with sequences which are highly repetitive, then you would end up with sequences at the leaves of those trees, which as a function of your substitution model, if you you would have certain differences. But if you start off with repetitive sequences, then you would certainly be able to end up with many instances at the leaves of those repetitive sequences that further undergo changes due to the evolutionary history of the tree. So in summary, like our model this framework only generates trees. It does not, and you can then, using features such as SeqGen, you can generate sequences down that tree. But the tree itself is not constructed with any input regarding sequences itself. The tree is constructed independently.
Okay, so independence of the trees with the evolution of sequence, actually.
Yeah.
So there is I will come back to your answer, but there is another question again by Maxime Senkov from University of Dundee. So Maxime is asking, how would the program handle PFAM clans where multiple protein domain annotations have sequence similarity, possibly due to convergent evolution? Would it be able to incorporate a priori knowledge and have two trees that eventually join up?
So two domain trees that eventually join up. How would the program where multiple domain trees have sequence similarity, possibly due to conversion, would be able to incorporate a priori knowledge, have two trees that eventually join up? So it would not be possible to combine two trees. But it would certainly, you could restrict the, actually, in summary, no, you would not be able to join up two protein domains into one, I guess, conversion domain in that sense, because the trees themselves are evolved independently. So like the evolution of one domain tree is completely independent of the evolution of another domain tree as it stands currently. But both domain trees can evolve within the same gene trees. So that's a feature that is currently supported. But yeah, the trees themselves at any given level. So at the level of gene trees, with regards to multiple gene trees evolving down a species tree, the evolution of each individual gene tree is independent of other gene trees. And similarly, at the domain level currently, the evolution of each domain tree is independent of other domain trees. But you can select which set of gene trees it evolves down. And so it can evolve down two sets of gene trees which are, for example, relevant for a particular domain or a set of domains.
Okay. Thank you for the answer. So there is also another question from Crystal. She's asking about the availability of the tool.
So this tool is available through the website of our lab, so the UConn CompBio lab. So if you go to the to UConn CompBio lab, if you Google, or if you look up Mukul Bansal UConn, you should be able to reach the UConn CompBio lab, and there's a page on that on that website which essentially lists all the different software packages as part of the lab that is developed by the lab, including like the range of DTL, which we discussed here, and in there should be SaGePhy, which you can essentially download as a software package from there.
Okay, so I'm not reading all the questions in the order, but so there is another question from Prabhav Kalagati from Max Planck Institute in Germany. So he's asking, I'm concerned about how you allow evolution of GC content, which is caused by GC biased genes conversion and deamination of 5MC to T. So SecGen only allows stationary models like GTR and no GC evolution. So I think this question is also related to the evolution of sequences.
Yeah. So yeah. Again, I think I guess the trees themselves are independent of the sequence models, but you can and for evolving sequences in this scenario, we used SeqGen to essentially test whether or not we could do this. But in theory, once you have these different trees, you can use any model that takes advantage of existing trees to then evolve sequences down that. So the simulation framework, as it stands, is independent of sequence models for generating trees. It simply uses this birth-to-death process to create or destroy lineages, and you can then evolve your favorites. You can use your favorite sequence generator or sequence model to then evolve sequences down that given tree.
Okay, thank you. So next question is by Maria Rosa Domingo Sananes from Nottingham Trent University.
Mm-hmm.
So she's asking, how computationally expensive are the simulation? Could you do genome level simulations? That is a single species tree and many gene families with variable parameters.
Yeah. So actually, this framework is not very computationally expensive at all, based on at least my experience from running this. In that, you could, like, in terms of at least some of the benchmarking that I've done with regards to generating multiple species and gene trees. We generated hundreds of species trees and 300 gene trees, for example, to do these different experiments with looking at what set of different events do we produce, and for doing the benchmarking with the DTL reconciliation framework. And of course, I'm sure Professor Bonsall can speak more about using this framework in the last 2 years on work that has been done since in the lab using this. But yeah, certainly you can use in terms of genome level simulations, you can evolve as many gene trees as you want corresponding to the different genes in the genome, and they could all evolve down a single species tree. And these different gene tree evolutions are independent of other runs. So if you really wanted, you could do these in parallel. And I would generally, the framework itself is fairly efficient in the sense that you could easily generate thousands of these within a reasonable time frame.
Okay, thank you. So these are all very interesting questions and answers also. So I will continue with the relation with the evolution of sequences. So finally, if you make a sequence evolve along the branches of the tree that you have simulated, you end up with a single sequence for each leaf of your trees. Is that correct?
Yes, that's exactly what you would have. Yeah. Mm-hmm.
Okay. Would it be possible to use it, for instance, to simulate the evolution of multiple transcripts? I mean, say you have the evolution of genes and you have the evolution of the domains that are within these genes. Can you use that also to simulate the evolution of alternative splicing, for instance?
Yeah, that's an interesting question. I'm not exactly sure how, that you would enforce, I guess, the alternate transcript. Like, I guess you would start off with the same initial sequence, and you would look at how multiple runs of the how multiple bootstrapping of passing that sequence through the tree, results in different outcomes. I'm not sure to what extent that would capture events like alternative splicing, in the sense that, the methods, the tree itself is not aware of exons or introns and all these things. So, but I can certainly see an opportunity to build on top of this to support these different features, because as it stands, the feature set is fairly it's a fairly simple construct where you simply have different trees that are subjected to these evolutionary events, and now what you do with them is fairly wide open in terms of how how you, for example, choose to what kind of questions you choose to answer given these tree phylogenies. So so yeah, just like I think one sequence can be evolved down it, I'm sure you could have multiple, I guess, transcripts evolved down that. But I guess some of the details would have to be informed with domain with that specific expertise, on that specific problem. So, but yeah, I mean, it's certainly like there's a lot of things you can extend this framework to.
Yeah, I agree. It's very interesting. Thank you. And so also, I will go back to the question of Maria Rosa Domingo Salanes. She was asking if it was possible to genome level simulation, so to simulate the species tree and many gene families actually that correspond to this species tree. So my question is, would it be possible? So, because more and more studies are looking at the evolution of genes in centenies, actually. So when the genes are closer in the genome, so the gene trees will look like each other. So would it be possible to integrate such information in the generation of the gene trees? So close gene trees whose genes are actually close in the genomes, on the genomes.
That's yeah, that's actually interesting. I mean, I would imagine you could. Yeah, that's a good question. I mean, obviously, I've not thought about this before, so I don't have any don't have a ton of intuition on this. But I would imagine you can certainly do certain things like if you wanted to enforce like similarity in terms of different gene trees evolving down a species tree, you could do things like let's say in our framework, for example, you have this notion of biasing the start of the gene tree within the species tree, right? So you could, for example, for a specific group of genes, you can enforce, or you could bias the location of the start by using these weighted probabilities of these different nodes in the species tree. You can bias the start of that group of genes at a specific location within the species tree, and then in that way it would by biasing the start location, you would somewhat bias the similarity of that group of genes evolving down the species tree, which could be one way in terms of kind of making sure that these group of genes have a somewhat similar topology and somewhat similar set of events. But in terms of controlling the stochasticity of which set of events get sampled, I think you could kind of control for similarity there by when choosing parameters for the duplication, transfer, and loss rates, you can enforce, you can set those rates such that those group of genes that you want to be similar would have similar rates, while a different set of genes would have different rates, which would essentially tweak the number of, let's say, transfer or duplication or loss events to be similar across that subset of different gene families. So I guess those are one possible way to do it, but yeah, certainly would require some thought.
Thank you.